Hello students, I am Rekha from Government Science College, Bangalore. So today the topic of discussion is the translocation of organic solutes. So we shall study the introduction of this chapter, path of movement, evidences, vein loading and unloading. So coming to the introduction of the topic, the movement of organic food materials or the solutes in soluble form from one place to another in higher plants is called as translocation of organic solutes. So this translocation or transportation always takes place from the region of higher concentration to a region of lower concentration. That is from the supply end to the consumption end. Translocation is essential in higher plants because in higher plants only the green parts of the plant, that is the leaves, it undergoes photosynthesis and the remaining part must be supplied by the leaves itself. And secondly, during the germination of the seeds, the insoluble reserve food material is converted into soluble form and supplied to the growing regions of young seedling till it develops its own photosynthetic system. So next coming to the path of movement. So how this solutes is transported from one place to the other place. There are two main translocatory tissues present in the plants, namely the xylem vessels and the phloem sieve tubes. So both these tissues differ morphologically and physiologically. The xylem vessels are dead when mature and consist of only the cell walls, whereas the sieve tubes are living. So let us study the comparative study of the xylem vessels and the phloem sieve tubes. So first coming to the anatomical comparisons, vessels have the open ends, whereas in sieve tubes, at the ends of the sieve tubes, you see the presence of sieve plates. Xylem vessels are wider and the sieve tubes are very narrow. Vessels have a thick, rigid, lignified wall, whereas the sieve tubes have thin and more extensible cellulosic walls. So, coming to the physiological comparisons, the xylem vessels are dead when mature but functional. The sieve tubes are alive when mature and they are functional. Vessels are permeable to both solute and solvent. The sieve tubes are semi permeable. Vessels have low sap concentration. As a result, they do not have the turgor pressure. They absorb water or air when it is cut and they translocate both solute and solvent. The sieve tubes have high sap concentration. The turgid cells have high turgor pressure. They exude the cell sap when it is cut and they translocate only the solute. So, coming to the evidences to support the translocation of organic solutes in phloem are, first one is exudation from the incision in bark. When an incision is made in the bark of a deciduous tree, there is an exudation of liquid containing sugars in high concentration. A careful examination reveals that the exudate comes from the sieve tube elements. The second evidence is the ringing experiment or the girdling experiment. So this experiment have provided sufficient evidence. The ringing of phloem breaks the downward movement of carbohydrates or the organic solutes. So this ringing of phloem, it causes a marked accumulation of sugars above the ring and a marked decreased below the ring. So it can be explained as the downward translocation of carbohydrate and other organic compounds occurs through the phloem tissue. The third evidence is the analysis of sap from the aphid stylet. An aphid sucks the sap by penetrating its style portion of the mouth to an individual sieve tube. So during this, the feeding aphid is anesthetized by a gentle stream of carbon dioxide and its body is cut by a blade, leaving the severed stylet. So the tube connecting the contents of the sieve tube with the exterior, when it is cut due to the hydrostatic pressure in the sieve tube sap begins to come out through the aphid stylet. The droplets are collected and analyzed chemically, and the result supported that the translocation of solute takes place through the phloem sieve tubes. The fourth evidence is from the tracer techniques. Experiments were done on the leaf to photosynthesize in an atmosphere of carbon isotope C13 or C14 and observed the labeled sugar moved in the phloem. This technique has also shown clearly that sugar moves in the sieve elements rather than other phloem cells. The fifth evidence comes from the chemical analysis. Chemical analysis of phloem revealed that these are relatively richer in carbohydrate and organic nitrogen compounds as compared to the lower percentage in xylem tissue. Therefore, it is obvious that solute moves through phloem. So, coming to the structure of the phloem cells. The structure of phloem tissue is well modified for the conduction of solutes. So, phloem consists of the sieve tubes, companion cells, phloem parenchyma, the fibers, and sclerites. The structure of phloem consists of these four elements, and of these, the sieve tubes are involved in sugar translocation. Sieve tubes are elongated cells which are arranged longitudinally end to end. The parenchyma cells are closely associated with them and remain connected through fine cytoplasmic threads called the plasma desmata. Certain parenchyma cells, which are referred to as the companion cells, arise from the parent meristematic cells along with the sieve tubes. So, companion cells provide energy to the sieve tube and participate in loading and unloading of assimilates. So, coming to the direction of the phloem transport. So, the movement of the solutes in plants is of two types, that is linear as well as the radial. So, most of the translocation in plants is linear. So, migration of the solvents from upwards to the lower side takes place, and also the vice versa is also true. So, downwards from the leaves to the roots, and upwards in case of germinating seeds and underground perinating structures. Radial translocation takes place during fruit and seed development. So, what does the phloem consist of? So, phloem, the analysis of the phloem tissue revealed that the phloem sap contains sugars at a concentration of 16 to 25%, while the amino acids or the amine contains about 0.04 to 4%. The major carbohydrates are sucrose, stachyose, and raffinose. So, in addition to carbohydrates and amines, it also contains the other organic substances and inorganic substances. So, the next topic is vein loading and unloading, which is also called as phloem loading and unloading. The translocation of organic solutes takes place through the sieve tube elements of phloem from the supply end to the consumption end, that is, from source to the sink. Transfer of soluble sugars from the basophil cells to sieve elements in the phloem is called as vein loading, and the reverse process is called as vein unloading. The region which supplies the sugars to phloem is referred to as source, and where it is consumed is called as the sink. The sources are the green leaves, while the sinks are the growing points of roots and shoots, storage organs, stem, rhizomes, tubers, etc. So, what is phloem loading? The movement of sugars from the basophil cells to the sieve tubes of phloem may be either through symplast or apoplast. Symplast is cell to cell movement through plasmodesmata. Apoplast is cell walls outside the protoplast. Sugars are actively loaded from apoplast to sieve tubes by an energy driven transport located in the plasma membrane of these cells called sucrose H plus symport or co-transport mechanism. So this is the diagram which represents the sucrose proton model. So this model explains the transport of sucrose across the membrane. The membrane contains the enzyme ATPase, which hydrolyzes one molecule of ATP and pumps two protons out of the cell. This makes the outside more positively charged and the inside becomes more negatively charged. The sucrose proton translocator, in its negatively charged form, combines with a proton and a sucrose molecule and it undergoes conformational changes. Sucrose in this way move across the membrane and both the sucrose and protons are released into the interior of the cell. The translocator, after receiving the proton, restores its original conformation and gets again ready to combine with another sucrose molecule. So this process will continue until the difference in chemical potential of protons and sucrose on two sides of the plasma membrane become equal. So in the process, the sucrose proton translocator is a kind of symport or a co-transport system because there is a linked movement of the two entities across the membrane. Then what is phloem unloading? Phloem unloading occurs in the consumption end or the sink organs such as developing roots, tubers, reproductive structures, etc. Sucrose unloading also occurs through either symplast or apoplast. So symplastic movement takes place in meristems, roots and young leaves, and the apoplastic movement takes place in the storage organs such as fruits, roots and the stems. So, coming to the conclusion of this topic, translocation is the movement of organic substances from the site of its synthesis to the site of its utilization. The organic substances are transported in sieve elements of phloem which work closely together with the companion cells. The loading of sugars in the phloem at the source and their removal at the sink establishes an osmotic potential that causes the movement of sugars from source to sink. Thank you for listening my lecture.